Welcome to the Nordic Venture Show, your speed date with the coolest ventures, brightest founders, and exciting things happening in the Nordics. In this episode, our venture date is with Desentum, a clinical stage biopharmaceutical company focused on developing novel allergen immunotherapy AIT products.
Welcome to the deep dive. Today we're digging into source material about, well, something that affects a huge number of people around the world.
2 billion people, roughly. It's massive.
Exactly. We're talking about allergies, the seasonal sniffles, reactions to food, maybe sneezing around pets. If you've dealt with it, you know how disruptive it can be.
Absolutely. And it's not just inconvenient, right? For a lot of people, it's a serious daily burden. Chronic symptoms, always worrying about a severe reaction.
Yeah, and dealing with it usually means, what? Trying to avoid triggers, which isn't always easy, or possible even.
Right. Or you rely on medications, antihistamines, sprays, that sort of thing.
And those just manage the symptoms. They don't actually fix the underlying problem.
Exactly. They're just dampening the reaction after it starts. They don't change how your body is reacting in the first place.
So the only thing currently available that does try to tackle the root cause is something called allergen immunotherapy, AIT.
That's right. Traditional AIT.
Okay, immunotherapy. Sounds pretty involved. What's the traditional way look like?
Well, the idea with traditional AIT is to basically re-educate your immune system.
Re-educated how?
By giving really small, carefully controlled doses of the actual natural substance you're allergic to. The pollen or the dander extract.
Actual allergen.
Yep. Usually as injections or sometimes drops under the tongue, and you gradually increase the dose over time.
To sort of build up tolerance.
Exactly. The goal is to desensitize your body, to shift your immune response away from that allergic reaction and towards tolerance.
And I guess if it works, it can be life-changing. Fewer symptoms, maybe less need for medication.
Definitely. It can have sustained effects even after you stop treatment. The potential benefits are huge.
But there's always a but. The source material points out some pretty big downsides to the traditional method.
Oh, yeah. It's a massive time commitment. We're talking 3 to 5 years of regular treatment.
3 to 5 years, wow.
And the results, they can be unpredictable. Works great for some, not so much for others.
And the really big one, I suppose, is the risk, since you're using the actual allergen.
Precisely. There's always a risk of triggering an allergic reaction during the treatment itself, sometimes even severe ones, like anaphylaxis.
So you need careful supervision in a clinic. Adds complexity.
It does. And for some really tough allergies, like food allergies, traditional AIT isn't always available, or maybe it doesn't work long-term after you stop. So it's often still just avoidance and carrying an EpiPen.
Okay, so that sets the scene. Allergies are everywhere. The main treatment targeting the cause is long, risky, unpredictable, sometimes not even an option.
Yeah, it's a real challenge.
Which brings us to our deep dive today. We're looking at source material about a totally different approach. A company called Desentum is developing a new kind of AIT.
A clinical stage biopharma company.
Sure. And their whole mission is to make AIT safer, maybe much faster, and more reliable by, well, fundamentally rethinking the key ingredient.
Instead of the natural allergen, they're engineering something new. It's really applying some cutting-edge science here.
Okay, let's unpack this. First, maybe a quick refresher on what's actually happening in the body during an allergic reaction. What goes wrong?
Right. So think about your immune system encountering something harmless like pollen. Normally, certain immune cells, these naive T helper cells or Th0 cells,
They're like the first responders.
kind of, yeah. In a healthy response, they get directed down a path called the Th1 pathway.
Th1, okay.
And that leads to making a type of antibody called IgG, immunoglobulin G.
And IgG is generally good, right? Protective.
Exactly. IgG recognizes the pollen, helps clear it out without causing any fuss, no symptoms, your body just handles it.
But if you're allergic,
Yeah,
the system takes a wrong turn.
precisely.
Yeah.
Same pollen, but in an allergic person, those Th0 cells go down the Th2 pathway instead.
Th2, not Th1.
Right. And Th2 leads to making a different antibody, IgE, immunoglobulin E.
IgE. That's the culprit in allergies.
That's the one. IgE loves to stick to mast cells. These are immune cells sitting in your tissues, skin, airways, gut.
So the IgE antibodies just latch onto these mast cells.
Yeah, they basically coat the surface, arming them, like setting little traps.
Okay, so the mast cells are primed. Then what?
Then, the next time you encounter that same pollen, the pollen particles land on these armed mast cells. And if a pollen particle connects two or more IgE antibodies together,
It bridges them.
exactly, that cross-linking is the trigger. It's the signal for the mast cell to just dump its contents.
Histamine and all that other inflammatory stuff.
All of it floods the area, causes the itching, sneezing, swelling, all the allergy symptoms, and in bad cases, it can be systemic, leading to anaphylaxis.
Wow, okay. So the core issue is IgE on mast cells getting cross-linked, triggering histamine release.
That's the mechanism.
And traditional AIT tries to sort of push the system back towards the IgG path using the natural allergen over years, despite the risk.
Yeah, it's trying to shift the balance, but using the very thing that causes the problem.
Which brings us back to Desentum. They spun out of research in Finland back in 2011, VTT in University of Eastern Finland. Sounds like serious science background. And they said, okay, what if we don't use the natural allergen? What if we engineer a better version?
That's the core idea. Use modified recombinant hypoallergens.
Hypoallergen, meaning less allergenic. How do they make it safer?
This is the clever part. They use really targeted changes to the protein's structure. Specific modifications designed to make it much harder for the protein to cross-link those IgE antibodies on the mast cells.
Ah, so they're tweaking the molecule to stop it from pulling that trigger.
Exactly. Less IgE cross-linking means much less histamine release, so a lower risk of reactions during the immunotherapy itself. That's the safety angle.
Okay, makes sense. But it still needs to teach the immune system the right response, right? It needs to generate that protective IgG.
Absolutely critical. So the modifications are designed very carefully to preserve the protein's overall natural shape, its native conformation.
So it still looks enough like the real thing to the rest of the immune system.
Precisely. It can still effectively stimulate the parts of the immune system that generate the good antibodies, the IgG.
So you make IgG against the hypoallergen.
And then, when you encounter the natural allergen out in the world, that new IgG you've made runs interference. It intercepts the natural allergen, blocks it from binding to the IgE on your mast cells.
Okay, so it's like sending in a safe training dummy that teaches your body to make defenses that work against the real threat.
That's a great way to put it. You get the immune re-education, but hopefully without the risk during training.
And they also mentioned a novel formulation for controlled release and recombinant production.
Yeah, the controlled release is another potential safety feature. And recombinant production means they make the protein using biotechnology.
Not extracting it from natural sources. Why is that better?
Consistency and quality. And natural extracts can vary a lot. Recombinant production means every batch is identical, pure, high quality. That's essential for a drug. It's made to GMP standards, good manufacturing practice.
Got it. Yeah. So engineered safety, still effective at triggering the right immune response, controlled delivery, consistent production. And the big promise is fewer injections, maybe a much shorter treatment. That 10-week course mentioned in the source material sounds amazing compared to years.
It would be a complete game changer for patients, going from years to weeks. That's huge.
But making these things, designing them just right, sounds incredibly complex. The sources mentioned some advanced tech they use.
Oh, definitely. This relies on some powerful tools, like mass spectrometry, specifically native mass spectrometry.
Mass spec measures the weight of molecules, basically. What's native mean here?
Standard mass spec often unfolds or breaks up proteins. Native mass spec lets them look at the proteins, even big complexes, while they're still folded in their natural shape.
Keep them intact.
Yeah, it's super sensitive, helps them check the exact structure, how it folds, how stable it is, and crucially, how their modified hypoallergen actually interacts with IgE antibodies at a molecular level.
So they can confirm it's not binding IgE as strongly.
Exactly. Verify the design works. They also use protein crystallography.
Getting the 3D structure, like a detailed map.
The ultimate map, yeah. You crystallize the protein, shoot X-rays at it, and figure out where every single atom is. How the amino acid chain folds up into helices and sheets, the overall 3D shape, even how multiple protein chains might fit together.
And knowing that structure lets them be really precise with the modifications.
That's the key. It allows for rational design. You know exactly where to make changes to disrupt IgE binding, but preserve the parts needed to stimulate IgG. The source mentions foundational work by Juha Ruvinen, finding the first allergen IgE binding site structure. That kind of knowledge is crucial.
Fascinating. Using atomic-level blueprints to engineer safety. Okay, so they design it, check the structure. How do they test if it actually causes less of a reaction biologically?
Good question. They use functional tests. A T1 is the histamine release assay, or HRA.
Histamine release.
Yeah.
Using actual cells.
Yeah, they use basophils, which are very similar to mast cells. They get them from blood samples of allergic patients.
Okay. And then
They expose these cells to the natural allergen and then separately to their hypoallergen candidate. And they measure how much histamine gets released.
the goal is much less histamine with the hypoallergen.
Exactly. If it triggers significantly less release, it confirms it's behaving like a hypoallergen in a relevant biological system. The sources mentioned they work with Reflab in Denmark for this.
So structure tools to design and check, functional tests to confirm the biology. It's a really integrated approach.
It really is. It's not just tweaking the old method, it's a complete redesign based on understanding the protein and the immune system deeply, engineering the molecule to push the immune response down a better path.
All right, the science sounds solid. So where are they with actually getting this to people? They have clinical trials ongoing.
Yes, they're a clinical stage. Their lead candidate is called DM101PX, and it's for birch pollen allergy.
Birch pollen, huge one for many people. What have the trials shown for DM101PX?
They ran a phase 1 study first, looking at different doses, mainly checking safety and tolerability in adults with birch allergy. And
It was found to be safe and well tolerated. They also saw positive signs in immune markers, things like IgE and IgD levels, suggesting the desired immune response was starting.
okay, safe is step 1. What about testing that short treatment course?
That was the next study, DM101-C002. They specifically tested short courses, just 10 injections over 10 weeks.
10 weeks, that's the target timeline. How did that go?
Also safe and well tolerated, and really importantly, it showed a strong increase in allergen-specific IgG4 antibodies.
IgG4. That's a specific type of IgG linked to tolerance immunotherapy, right?
Exactly. It's considered a key marker of success for AIT. It plays a big role in blocking those IgE reactions, and they saw a robust IgG4 response after just 10 weeks.
Okay, great. But did those antibodies actually work? Did they block the allergic mechanism?
Yes, they tested that too. They took antibodies generated in the trial participants and used them in lab tests, looking specifically at whether they could block IgE from activating basophils.
And could they? The results showed they were highly efficient at it. The antibodies induced by the 10-week treatment could effectively stop those allergy cells from getting activated.
That's really strong evidence, connecting the immune response to the actual desired effect. And this was done in Canada, partly using a pollen exposure chamber.
Oh, it is, yeah. Which gives you more confidence that the effects seen might translate to real-world exposure.
Okay, so 10-week course, safe, generated the right kind of protective antibodies, and those antibodies showed they could block the allergic reaction pathway. Sounds very promising for birch allergy. What's next for DM101PX?
Phase 2 development is planned for 2025. That means bigger trials, looking more closely at efficacy and finding the best dose.
Exciting. And birch is just the start, presumably. This technology seems like it could work for other allergies too.
Absolutely. The source confirms they have a pipeline. Each allergy needs its own custom-designed hypoallergen based on the specific protein involved.
What else are they working on?
Peanut allergy is a big focus. They've made modified versions of ARA-H2, a key peanut allergen. Those are in preclinical testing now.
Peanut. That would be huge.
Massive unmet need. They're targeting clinical trials for peanut in 2026. They're also working on grass pollen, looking at modified FALP1, and cat allergies are also in preclinical development.
Wow, covering some major ground. And we've seen some recent news reflecting this momentum.
Yeah, they're actively sharing their results. They're presenting the DM101PX clinical data and some peanut findings at the big EAACI Allergy Congress in June 2025. And they reported positive phase 1 data back in April 2025 as well.
Plus, they raised 12 million euros early in 2024, and the CEO got an honorary doctorate. Signs of progress and recognition.
Definitely suggests confidence in their approach, both from investors and the scientific community.
And that scientific foundation seems really strong coming out of VTT and University of Eastern Finland.
Absolutely. Deep expertise in the core science. And they've built a team with experience across the board, biotech leadership, R&D, clinical trials, manufacturing, regulatory, all the pieces you need.
And they're smart about collaborating too, right? Using external experts.
Yeah, the sources highlight that. VTT for some research production, University of Eastern Finland for that specialized mass spec, Reflab for the histamine assays, Medical University of Vienna for mouse studies, and Biovian for the actual GMP manufacturing of the drug.
Tapping into a whole network. Makes sense for such complex work.
It's a really efficient way to access top-tier capabilities.
Okay, let's bring this home. Why should you, the listener, care about this deep dive? If you're dealing with allergies, what's the potential impact here?
Well, it matters because this could be a fundamentally better way to treat the cause of allergies, not just masking symptoms, not just enduring years of risky treatment.
A potentially safer, much faster, and maybe more reliably effective long-term fix.
That's the goal. Using these engineered molecules to basically guide your immune system towards tolerance, safely and efficiently.
And those early results for birch pollen, strong protective antibodies, blocking the allergy cells, all after just 10 weeks, that's got to be exciting news for allergy sufferers.
It's definitely promising. Of course, it's still investigational, more trials are needed, but the core idea, engineering the allergen itself for safety and efficacy, is really powerful.
So the key takeaway seems to be Desentum's using smart science, combining protein engineering and immunology to try and create a much better version of allergy immunotherapy, safer, faster, based on these recombinant hypoallergens.
Yeah, what really stands out is that rational design, using the structural and functional understanding to build a molecule that avoids the bad stuff but triggers the good stuff in the immune system.
And thinking bigger picture, if you can engineer molecules to precisely redirect the immune response away from allergy, could that kind of approach work for other things?
That is a really interesting thought. Other conditions where the immune system is maybe overreacting or attacking the wrong thing, like certain autoimmune diseases.
Could this precision immunotherapy be a platform for more than just allergies down the line? Something to definitely keep an eye on as the technology evolves.
Thanks for tuning in. You can follow us on Spotify, Apple Podcasts, Google Music, or your favorite app, and connect with us on LinkedIn for more stories and insights.